Yes, let's continue the locomotion and reproduction in protozoa. We are in the module 15. In this module, we are going to learn about the transverse binary fission of parameecium. Here in this module, we are going to learn about the transverse binary fission of parameecium here. Let us see the transverse binary fission of parameecium. As I said to you, longitudinal binary fission in euglena and transverse binary fission in parameecium. These two are very, very important here in this particular state examination aspect. So, transverse binary fission is performed by the parameecium. The parameecium is commonly called as slipper animalcule. Parameecium is commonly called slipper animalcule. Let's take a look on this particular structure of parameecium. It has a flat oral surface and a convex aboral surface. That means on oral surface it is flat and aboral surface that it is going to be convex. Oral surface means this surface we say oral and this surface we say aboral. Okay, the oral surface bear oral groove that opens through cytostome and into the cytopharynx. Cytostome and cytopharynx. So then let's continue here that the oral groove that opens into the cytostome. Let's see the oral groove here in the parameecium, the how it is. So that place which we are showing here, that is oral groove and cytostome, where oral groove opens into the cytostome, then cytostome opens into cytopharynx. So that we, as we know that ectoplasm contain infraciliary system. Did you remember? We learned that in locomotion in protozoa. Ectoplasm contain infraciliary system and protective organelles called as trichocysts are present here below the pellicle in the ectoplasm. Nowhere related to with our reproduction, just by namesake we are saying that. Okay, then when it comes to the endoplasm, contain two nuclei of different shape, size, and function. What are the two nuclei? Macronucleus and micronucleus. So, by the chromosomal aspect, we say macronucleus as polyploid in nature and micronucleus is diploid in nature. So that you may know that haploid means single set of chromosome. Two sets of chromosome if present, we say diploid. Multiple sets of chromosome if you present, we say that condition as polyploid condition. So okay, then let's come through this. That contractile vacuole. When we talk about the contractile vacuole, as we said in euglena, there is only one contractile vacuole is present. Now we'll see the two contractile vacuole, anterior contractile vacuole and posterior contractile vacuole. So then one is in anterior position and other is in posterior position. So the many food vacuoles are also present in case of parameecium. Many food vacuoles are present. So this is the parameecium, the slipper animalcule. Parameecium is considered as slipper animalcule. Okay. So why? Because see the shape of the parameecium, slipper. So that body covered by pellicle, that pellicle contains cilium. So below the cilium, you can see small stinging structures. We say them as trichocysts. They are used for protection. Here, body is covered by pellicle. Then you can see the contractile vacuole there. One is an anterior side. We call it as anterior contractile vacuole. And some radiating canals are present here. That no matter with radiating canals for us. So then here the cilia and see that oral groove. Oral groove opens into the cytostome, then into the cytopharynx. In that way, you have to look forward. So actually, we learned the parameecium binary fission on board yesterday. So I think I'm watching some new people entered here. So we do one thing. Let me complete the parameecium binary fission first of all. Then after I'll go to the board and I go for the binary fission of parameecium again. So that means in every topic I use it to explain in two steps. One I'm explaining on the board, and after that we are coming with the animative portion of this particular. That means this is stage 2. But some people entered today. So after let me complete this. Then after I'll go back on board, and again I'll try to explain the binary fission and parameecium and euglena binary fission. So let me complete this first of all. Let me complete this module. Then after I go back again, I'll explain the parameecium binary fission to you also. The micronucleus and macronucleus. There are two nuclei here. We say nuclear dimorphism, that's such condition. If any organism is having two nuclei and they are in different shapes, we call that condition as nuclear dimorphism. Okay. So then one is smaller in size, that you can say that is micronucleus, and the bean-shaped nucleus we call macronucleus, showing that mouth of the cell that we say cytostome, which another one we are showing the anal pore, cyto, that the gullet, cytostome opens into the gullet, then it opens into the food vacuole is going to be formed there. You can see not much necessary all these particular issues for us, just for general knowledge sake we are here. What we need to learn, that we already learned yesterday, that food vacuole circulating you can see in the particular. So the cytoplasm, one more showing to you. So this is a parameecium, that basic structure of the parameecium. The things to be remembered over. What need to remember? Parameecium body is covered by pellicle. So let me just follow the arrow mark here. Parameecium body is covered by pellicle, and there are two nuclei, macronucleus, micronucleus. And there are two vacuoles, anterior contractile vacuole and posterior contractile vacuole. There is an opening, we say oral groove, which opens into the cytostome, then cytopharynx. These three are enough here for us to learn the binary fission, because we're not going to for anatomy of parameecium, just to some basic issues of the parameecium, and now we entered into the binary fission. So before undergoing binary fission, the parameecium stops feeding and its oral groove disappears. So the micronucleus divides by a process called mitosis. Macronucleus divides by amitosis. Please don't confuse with amitosis, nothing but not like that meiosis. Meiosis is the division which occurs in sexual reproduction. So why I am saying here, because the people that in SSC or earlier classes you may learn over, there are two types of division, mitosis and meiosis. If it is not mitosis, they feel so that themselves they are, it has meiosis. Not like that, not like that. Equal division we say mitosis. The unequal division of the macronucleus, they are going to say it as amitosis. So what I am saying, that is not meiosis, that is mitosis itself. Mitosis, what it means? The equal division of the nucleus, asexual multiplication of nucleus. Amitosis, unequal division of the nucleus. So not similar with meiosis. Okay. So two oral grooves will disappear here. First of all, that parameecium is said the stop feeding. Then first of all, the karyokinesis occurs. We have two nuclei. We said the macronucleus undergo amitosis, micronucleus undergo that mitosis. Then after nuclear division, a constriction appear in the middle of the body, which deepens and divides the parent cell into two daughter individuals. The anterior daughter individual is called proter, and posterior daughter individual called opisthei. The proter receive the anterior contractile vacuole, cytopharynx, cytostome from the parent. And what about the opisthei? The posterior contractile vacuole and new oral groove is appeared in the proter new. Then opisthei. The opisthei receive the posterior contractile vacuole of its parents. It develops a new anterior contractile vacuole, cytostome, cytopharynx, and a new oral groove. Okay, so definitely it is going to be a confusing thing to the people who entered today itself. For those people, I will explain back in coming 5 minutes. I'll take a board and I'll go for that back again for you. The binary fission is completed in 2 hours. The parameecium will go 4 times binary fission a day. That one day. That means 16 individuals can be formed. For after the 4 divisions, a 16 number of individuals can be formed. Then the transverse binary fission of parameecium is also called homothetogenic fission, because the plane of fission is right angles to the longitudinal axis of the body. We said this is transverse binary fission. So that's a plane of fission is right angles. So it is right angle to the that particular kinety also. Did you remember that kinety we learned last week? that right angle to the kinetic, longitudinal kinesia, perpendicular to the transverse binary fission of paramecia. Okay. So just let me show once again here this particular portion. Anterior contractile vacuole, then the pores of the new contractile vacuole, the posterior contractile vacuole, the cytostome, cytopharynx, you can see there. So this is the transverse binary fission. See what's happening here. That particular nucleus is dividing, macronucleus by amitosis and micronucleus by mitosis. The one of the daughter is retaining the parental cytopharynx. So what other daughter is doing? Other daughter is going to form the new cytopharynx. Here in this case, you can see micronucleus dividing by mitosis, not amitosis. That was a spell mistake here. And another daughter individual, you can see a new cytopharynx it is going to form. So transverse constriction you can see here. So the posterior contractile vacuole, the feeding apparatus. Okay, that the macro and micronucleus division, the transverse constriction you can see clearly here. So that what happens to that particular rotor and opiste? Okay, so let me close this and I'll take the board here. I'll go for parameecium binary fission. Back again, some new people entered today. For that particular people, I'll go with the parameecium binary fission. Further, I'm taking board. Let me take the screen here. Okay, now we are entered in the screen. Let me start that particular position as earlier second year class. Now, let me introduce fresh that particular parameecium. We said parameecium is commonly called slipper animalcule, isn't it? See the shape of the parameecium. Parameecium is commonly called slipper animalcule. Body is covered by pellicle. So we can say this as pellicle here. Body is covered by pellicle. Above on the body, you can see the smaller cilia that 10,000 to 14,000 cilia are present on the surface of parameecium. So these are the cilia. Okay. So nearly 10,000 to 14,000. And see this area. This is called oral groove. Oral groove opens into cytostome. Let me write all the things to you. Oral groove, and oral groove opens into cytostome. That cytostome. Then cytopharynx. You can see the cytopharynx there. Okay, that opens into the reservoir. No matter for reservoir for us. Now let me draw the two nuclei here. A larger nucleus is called macronucleus and the smaller nucleus is called micronucleus. Then cytoplasm is getting divided up of two, outer ectoplasm, inner endoplasm. The border I am drawing here. Okay, so that this particular outer ectoplasm we can say, and the inner portion is said to be endoplasm. This is endoplasm. Okay. Then next, what next? What we need to see that contractile vacuole. We have two contractile vacuole. One is an anterior side, one is on posterior side. Anterior contractile vacuole and posterior contractile vacuole. These are enough actually to learn the binary fission of parameecium. These things are enough for us. That means these things to be in mind. Okay, the body is covered by pellicle and there is an oral groove which opens into the cytostome, then cytopharynx. Then there are two contractile vacuole, that anterior contractile vacuole and posterior contractile vacuole. Here writing on the screen is little complex here. Yeah, this is a posterior contractile vacuole. Okay, so anterior contractile vacuole and posterior. It will take time here actually to make a type like this, posterior contractile vacuole. Okay, I hope you understand my writing. Otherwise, we need to do like this for every labeling. It's very difficult here to go uh, typing like this. So I hope you understand. Yeah, this is posterior contractile vacuole. These are enough actually. That anterior contractile vacuole, posterior contractile vacuole, body is covered by pellicle that contains cilia, that outer ectoplasm and inner endoplasm. Then the two nuclei are there, the macronucleus and micronucleus. Okay, so let's start the paramecium binary fission again here. What we said earlier, that the paramecium binary fission occurs in favorable condition. Favorable condition. Binary fission is the process which generally occurs in favorable condition. What are favorable conditions? Favorable conditions are like this: availability of the food. The food should available for the organism, abundant water, availability of food, abundant water, optimum temperature, abundant water and optimum temperature. That can be considered as favorable conditions. So during favorable condition, the paramecium feeds and grows. The paramecium feeds and grows, then it starts the binary fission. Any binary fission, either euglena or paramecium. The binary fission include two stages, two steps, we said, that binary fission. Number one, karyokinesis, division of nucleus. We say karyokinesis. Number two, cytokinesis, division of cytoplasm. Division of nucleus is called karyokinesis and division of cytoplasm is called cytokinesis, we can say. So, as I already said to you, there are two nuclei in paramecium, isn't it? There are two nuclei. So, karyokinesis means division of both macronucleus and micronucleus. So that's why we need to see how they are dividing. First of all, karyokinesis. Let me write the karyokinesis. And after that, what is karyokinesis? We'll write here division of nucleus. Karyokinesis means division of nucleus. Okay. So, how many nuclei are present here? The macro and micronucleus. So, first of all, let us see the karyokinesis, division of nucleus in macronucleus. So, how does that macronucleus appear? This is a bean-shaped macronucleus. Okay. So, just what happens here? This is the chromosomal content present in the macronucleus. So, nothing happens here. Just macronucleus elongates and divides. The macronucleus elongates and divides into two nuclei. That's all. That no special divisions here. We call such kind of division as amitosis. Macronucleus divides and form two nuclei here. That what you call it as amitosis. Okay. So then let us see how that micronucleus is dividing. The micronucleus. If you see that micronucleus with diploid in condition, this micronucleus, that as we said, this undergo mitosis process. What is mitosis? Mitosis means the micronucleus is having the nuclear membrane, isn't it? The nuclear membrane disappears. In micronucleus, nuclear membrane disappears. Chromatin material is going to be turned in the form of chromosomes. Okay, if you remember, you learned this in your earlier classes, mitosis, the prophase, metaphase. That means nuclear membrane is there actually, that nuclear membrane is disappeared. You can see here that chromatin material is condensed in the form of chromosomes, and that chromosomes are getting arranged on the equatorial plane, and these particular chromosomes are going to be pulled into the polar regions. Then after they form two nuclei, nuclear membrane reappearance. I'm not going to into the details of mitosis, that I already have a special chapter for mitosis. So just to see the difference here, just to see the difference here. If you see macronucleus, nothing happened like this. No disappearance of this particular the macronucleus. The membrane is not disappeared. No condensation of the chromatin into chromosome. Just the macronucleus elongated and cut into two nuclei. That's why we call this such kind of division as amitosis. When it comes to the micronucleus, all the protocol of mitosis is happened, like nuclear membrane disappearance, you can see, and chromatin is condensed into chromosomes. They are arranged on the equatorial plate, and we call this stage as anaphase, isn't it? If you remember, this stage can be called anaphase. Okay, so uh, that particular chromosomes are in the equatorial region, and that particular pulled into the two poles. So this kind of division we say mitosis. Okay, so simply what do you say during the binary fission of parameecium? That first division occurs is karyokinesis. There are two types of nuclei here. One is macronucleus. Macronucleus undergo amitosis. Macronucleus undergo amitosis. And what about the micronucleus? Micronucleus, if you take that undergo that mitosis, micronucleus undergo mitosis. So this is called karyokinesis. Okay. Next, what next? The second one is cytokinesis. Cytokinesis. Okay. So what is cytokinesis? Division of cytoplasm. Cytokinesis means division of cytoplasm is called as cytokinesis. Okay, so let us see how it appears. That cytokinesis. Let me draw the parameecium first of all to see this particular. So already nuclear division is happened. That two macronucleus and two micronucleus you can see here. Now at this time, the time of binary fission, the oral groove is going to be disappeared actually. The oral groove is disappeared and a constriction appears. With which constriction? Transverse constriction appears. The transverse constriction will continue. The transverse constriction will be continued like this. Okay. So the transverse constriction continued and that results the formation of two daughter individuals. That's why we call the such kind of binary fission as transverse binary fission. That result the formation of two daughter individuals. So we have to name the daughter individuals here, whatever form. The anterior daughter individual is named as proter. The posterior daughter individual is named as opiste. So anterior daughter individual named as proter and posterior daughter individual named as opiste. So after the formation of two daughter individuals are formed. Hence we call it as binary fission, isn't it? What is bi? Bi means two. A particular organism divides into the mother divides into two daughter individual. We call this as binary fission. Why you call it as transverse binary fission? You see, the constriction appear transversely. That's why we say it as transverse binary fission. Okay. Then after what we have to do, these particular two daughter individuals need to learn about some information about the two daughter individuals. So first of all, proter. Who is proter? After the transverse binary fission, in the form of the two daughter individuals, the anterior daughter individual is called as proter. Anterior daughter individual is called as proter. Now we should talk about, so what proter is obtained from mother. What it obtained from mother. From mother, what are the issues it obtained? That we need to talk about that particular. That from the mother, it obtained anterior contractile vacuole. Anterior contractile vacuole from the mother, as cytostome and cytopharynx of the mother is collected by itself. Anterior, the daughter individual is mother cytostome, mother cytopharynx is obtained by a proter itself. The nucleus is as usual, isn't it? That macro and micronucleus as usual. So now, what it formed new, that is the next thing to be seen. What it formed new, which is not collected by the mother. What it formed new? Posterior contractile vacuole. It formed posterior contractile vacuole. So that, let me see that particular. Here is the anterior daughter individual is called proter. So what I am saying, What it obtained from the mother? What it obtained from the mother? Anterior contractile vacuole, then cytostome, cytopharynx is taken. Macro and micronucleus is already distributed, you see here. So what is formed new? The newly formed issue here, this one, that posterior contractile vacuole. Okay. Then in the same way, let us see in the opiste also, that in opiste, first of all, we should write what is opiste. What is opiste? Posterior daughter individual. After the transverse binary fission of parameecium, the posterior daughter individual, which is formed here, considered as opiste. We named it as opiste. Okay. The posterior daughter individual is considered as opiste. Now, let us see what it obtained from mother. It obtained from mother is the posterior contractile vacuole. Obtained from the mother is posterior contractile vacuole. Now the question raises here, what it formed new? Newly formed structures are more in opacity, like anterior contractile vacuole, and that cytostome and cytopharynx are taken by their brother proter. So that's why mothers is taken by proter. That's why the cytostome and cytopharynx in the opisthe should form new. So what it form new? Anterior contractile vacuole, cytostome, cytopharynx. In both cases, we can write oral groove because in the mother itself we said oral groove is disappeared. So that's why the both cases that oral groove is going to be formed new. Okay. So this is the issue of transverse binary fission of parameecium. Let's come from the beginning here. That in the binary fission of parameecium, first of all, we need to learn some basics of the parameecium binary fission. What are the basics here? That body is covered by pellicle. The cytoplasm divide up of outer ectoplasm, inner endoplasm. That we need to take care about that. And anterior contractile vacuole and posterior contractile vacuole. There are two contractile vacuoles are present: anterior contractile vacuole and posterior contractile vacuole. And the opening, the small opening, we say oral groove that opens into the cytostome, then into the cytopharynx. Okay. Then the two nuclei are present here. We named that particular nuclei as the macronucleus and micronucleus. The larger bean-shaped nucleus is called macronucleus, and the smaller the nucleus is said to be macronucleus. The small spherical nucleus, we say it as micronucleus. Okay. So these are enough here to learn the binary fission of parameecium. And what do you say? This is micronucleus. Let me show here. This is the micronucleus. So these are enough here. Now let's move to the what we said, the wave, the binary fission of parameecium. Any binary fission occurs in favorable condition. What are the favorable conditions here? The availability of the food, abundant water, optimum temperature are considered as favorable conditions. Okay. Then the binary fission include two stages of here in the binary fission. One is karyokinesis and other is cytokinesis. What is karyokinesis? Division of nucleus is called karyokinesis. Division of nucleus. Here we have two nuclei, isn't it? For that, we need to use the plural sound. Division of nuclei. Nucleus is a singular one. Nuclei is a plural one. The division of nuclei is called karyokinesis here. The two nuclei are present here, as we know that one is macronucleus and other is the micronucleus. The macronucleus and micronucleus. First of all, let us see about the macronucleus. Just as what happens here, the macronucleus elongates. Such macronucleus is elongated and just it divides into two. We call such kind of division as amitotic division. Then micronucleus. Micronucleus follow the protocol like nuclear membrane disappearance, chromatin is condensed into chromosomes, and that particular chromosomes are equally distributed towards the poles, that we say mitosis. Exact distribution occurs in case of chromatin content. chromosomal content in case of micronucleus, but it won't happen in case of macronucleus. That we'll discuss in the next coming classes in the form of conjugation. Okay, so whatever macronucleus undergo a mitotic division, micronucleus undergo mitotic division. Next, what next? The cytokinesis. What is cytokinesis? Division of cytoplasm is called as cytokinesis. Division of cytoplasm is called cytokinesis. How does the paramecium is dividing? A transverse constriction appears. Hence, we call it as binary fission transverse. Transverse binary fission in paramecium. The transverse constriction appears and that continues, that divides the particular paramecium into two daughter individuals. What we said about the anterior daughter individual, proter, posterior daughter individual called opiste, posterior daughter individual called opiste. Then we need to distribute that particular issues between the proter and opiste. The anterior daughter individual proter from the mother, what it is collected from the mother, that anterior contractile vacuole, cytostome and cytopharynx. What it formed new? Posterior contractile vacuole and oral groove are getting formed new. Next, opiste. The posterior daughter individual is called as opiste. From the mother, it obtained posterior contractile vacuole. What it formed new? It formed new is anterior contractile vacuole, cytopharynx, and cytostome. This kind of distribution is required. That whatever after the completion of binary fission of paramecium, whatever the daughter individuals are present, they are completely similar, genetically similar. Why? Because just what you say, these kind of daughter individuals are not going for any meiosis. Sexual reproduction is not, they are not going for. Just asexual reproduction, any asexual reproduction. Remember this for both plants and animals, botany and zoology, biology. Any asexual reproduction, when the two daughter individuals are formed, that any number of daughter individuals are formed. We are talking about binary fission, that's why I said over two daughter individuals are formed. We call them as clones because both they look alike the same. The both are going to look like the same. Hence, we say clone. Tomorrow, when we see the longitudinal binary fission or any asexual multiplication, we say they are clones because they are completely similar genetically. That because their nucleus only divides and is distributed, isn't it? So that's why they completely look like mother and they are clones, we say. Genetically similar completely. Okay. Next, particularly for the paramecium. Okay, particularly for the paramecium. Throughout the last week, we are seeing that infraciliary system, isn't it? That infraciliary system is longitudinal in case of paramecium. That longitudinal. But how that our binary fission is present? That binary fission is transverse. That's why what do you say? The transverse binary fission of paramecium is perpendicular to the longitudinal kinesia. Hence, we called it as perkinetal fission. Need to define this as the transverse binary fission of paramecium is perpendicular to the longitudinal kinesia. Hence, we called it as perkinetal fission, or also called homothetogenic division. Such kind of division is considered as homothetogenic division. Okay, so this is the complete story of paramecium. As I already said to you, transverse binary fission of paramecium, longitudinal binary fission of euglena are very, very, very important in case of intermediate public examination. For NEET, if you remember the concepts of binary fission, that's enough. Because this is the chapter is having more weightage for the IP, locomotion and reproduction in protozoa. People who entered today, within a day, that means some new people are entered today. So within one day, we are going to complete this particular chapter actually, that locomotion and reproduction in protozoa. But soon we are going to start the new chapter after the completion of this particular. So at the time, you feel so little bit comfortable here. Or else, go for that. You can. These are individual parts here. You just don't feel anything new. The transverse binary fission of Euclid paramecium. I hope you understand this well, isn't it? I hope you understand this well because this is an individual topic, not related with, except that kinesia. Kinesia we learned earlier. So this is individual topic. So that's why I hope you understand this particular phenomena. So we continue with the classes here. In the next class, we'll go with the longitudinal binary fission of euglena. We'll see that back again, longitudinal binary fission of euglena, that in the form of 16th module, module 16. Let us try to answer some questions here at present. What paramecium is commonly called as? So paramecium is commonly called as slipper animalcule, isn't it? Slipper animalcule. The anterior daughter paramecium is called as clume, rotor, piston, flak. What do you say? So anterior daughter individual is called as proter. Anterior daughter individual is called as proter. The posterior daughter paramecium is called as, we call the posterior daughter individual as opiste. That's it. Thank you. That learned the parameecium binary fission today. We learned the parameecium binary fission. Thank you.